Hello, this is Sonali and my topic is the hydrogen bond and the structure of water. Hydrogen bonds. A hydrogen bond is a weak interaction that occurs when a hydrogen atom, which is covalently bonded to a more electronegative atom, that is oxygen or nitrogen, experiences an attractive force from another electronegative atom. In proteins and other biomolecules, hydrogen bonds are critical in stabilizing secondary structures like alpha helices and beta sheets, and they play a key role in the folding and the stability of the three-dimensional structure. The hydrogen bond is central to the structural integrity of macromolecules. In proteins, for example, hydrogen bonds form between the backbone amide and the carbonyl groups, which stabilize the formation of secondary structures. In nucleic acids like DNA, hydrogen bond between complementary bases, that is AT and GC, allow the double helix to maintain its stable structure. Talking about strength and specificity, while hydrogen bonds are weaker than covalent bonds, they are still significant because of they are highly directional. This specificity contributes to the precise folding and function of biomolecules. Multiple hydrogen bonds collectively provide substantial stabilization to biological structure despite their individual weakness. Now talking about water structure and hydrogen bonds, the structure of water is a unique aspect of biophysics because of its extensive hydrogen bonding network. Water molecules are capable of forming hydrogen bonds with up to 4 neighboring water molecules, aging to a dynamic and highly structured liquid. Now talking about tetrahedral geometry, each water molecule have a bent shape with the oxygen atom at the center and the two hydrogen atoms attached. The oxygen atom has two lone pairs of electrons which can form hydrogen bonds with nearly hydrogen atoms of adjacent water molecules. This results in a roughly tetrahedral geometry around each water molecule. This structure gives water its remarkable properties such as its high heat capacity, surface tension, and ability to dissolve a wide range of substances, making it an ideal solvent for biological molecules. Now talking about hydrogen bonding in water and biological systems. In water, hydrogen bonding results in cohesive network that influences its liquid state and the solubility of polar compounds. Water's hydrogen bonding is crucial for biological processes, particularly in maintaining the hydrogen trails around proteins and nucleic acids. This hydrogen cells stabilize the macromolecules and influence their folding and function. In proteins, water molecules often form hydrogen bonds with polar side chains, influencing the protein overall conformation and flexibility. The balance between hydrophobic interactions driving non-polar residues of the interior and the hydrogen bonds involving polar residues help to create the functional three-dimensional shape of proteins. In case of DNA, hydrogen bonds between base pairs, that is adenine, thymine and guanine cytosine, provides the primary stabilization for the double helix. However, these hydrogen bonds are also weak enough to allow the strands to separate during processes like replication and transcription. Now my another topic is helix coil transitions. The helix coil transition refer to the reversible process in which a polymer chain, like a polypeptide, transitions between an ordered helical structure such as alpha helix and a more disordered coil state. This transition is crucial in understanding the conformational changes of proteins and nucleic acids under different conditions. The transition between helix and coil states is governed by the thermodynamic principles. The helix state is more ordered and corresponds to lower entropy, lower energy configuration. In contrast, the coil state is disordered with high entropy but possibly high free energy. The balance between enthalpy, that is energy, and entropy, that is disorder, determines which state is more favorable under given conditions. Factors such as temperature, solvent conditions like pH or ionic strength, and the sequence of the polymer itself influence the transition. Now talking about hydrogen bonds. In the helical state, hydrogen bonds form regularly between the backbone atoms of the polypeptide chain, stabilizing the helical structure. As the protein transitions to the coil state, these hydrogen bonds are broken, allowing the chain to become more flexible. Van der Waals interaction and hydrophobic effects also plays important role in stabilizing the helical structure. In the coil state, these interactions are weaker or absent, leading to a more random configuration. Now talking about cooperative nature of the transition. In cooperative nature of the helix coil transition, when one part of a polypeptide chain forms a helical structure, it encourages adjacent part to adopt a similar structure. Conversely, when a portion of the helix unwinds into a coil, it becomes easier for neighboring regions to also transition to the coil state. This cooperative behavior is a hallmark of many biological transitions, where partial unfolding can lead to a cascade effect that propagates throughout the molecule. Now talking about mathematical models are used to describe the helix coil transition, including the Zim-Bragg model and the Lifson-Roig model. This model provides quantitative descriptions of how the transition occurs and allow predictions of the conditions under which a polypeptide will favor a helical or coil state. The Zim-Bragg model incorporates parameters that describe the initiation and propagation of helix regions of the chain, providing insights into the temperature and concentration dependence of the transition. Whereas the Lifson-Roig model is more detailed, accounting for the sequence-specific effects that influence helix stability, including the varying propensities of different amino acids to form helices. Now talking about biological implications, many protein contains helical segments, and their ability to switch between helical and coil forms is essential for their correct folding and function. Some enzymes rely on configuration changes, including helix coil transition, to regulate their activity or to bind substrates. Also, in nucleic acids, the helix coil transition can be important in the unwinding of DNA for transition or replication, with local helix coil transitions playing key roles in these processes. Also, various experimental techniques were used to study helix coil transitions, such as optical spectroscopy, that is, circular dichorism, and X-ray diffraction. These methods allow scientists to observe changes in the secondary structure of protein or nucleic acids in real time as they undergo transitions between helical and coil states. Now my next topic is protein globule and hydrophobic interactions. A protein globule refers to the compact three-dimensional structure that a protein adopts in its native state. This is often referred to a globular protein, which contrasts with more extended or fibrous proteins. In its globule form, the protein is biologically active and functions in processes such as enzyme catalysis, signaling, and molecular recognition. The formation of a globular structure is driven by the minimization of free energy, where the polypeptide chain folds into a configuration that is thermodynamically stable. This is the end result of complex molecular interactions, including hydrophobic interactions, hydrogen bonds, and van der Waals forces. Now talking about role of hydrophobic interactions in protein folding, one of the most crucial forces in the formation of the protein globule is the hydrophobic interaction. Hydrophobic interactions arise because nonpolar or hydrophobic amino acids residues avoid contact with water. In an aqueous environment, these hydrophobic residues tend to aggregate together, forming the core of the protein globule. These interactions significantly contribute to the collapse of the polypeptide chain into a compact form. Hydrophobic residues are typically buried inside the protein core, while hydrophilic water-attracting residues are exposed on the surface, allowing the protein to interact with the surrounding aqueous environment. Talking about energetic considerations, hydrophobic interactions are primarily entropy driven. When hydrophobic side chains are exposed to water, water molecules form an ordered cage around them, decreasing the overall entropy. By burying these hydrophobic residues in the core, the water molecules are released, increasing the entropy of the system, and thus making the folded globular state more thermodynamically favorable. The energetic balance between enthalpic, that is energy due to the molecular interactions, and the entropic, that is disorder, factors governs the final folded state. Hydrophobic interactions contribute to the entropic component, driving the protein into its native globule form. Talking about hydrophobic collapse, this is an early event in the folding process where hydrophobic residues come together, leading to a rapid decrease in the overall size of the polypeptide chain. This collapse drives the protein towards a compact globular state, which is then fine-tuned by more specific interactions such as hydrogen bonding and disulfide bridge formation. The core of the protein globule, consisting mostly of hydrophobic residues, becomes densely packed, stabilizing the overall structure. This packing excludes water from the core and further contributes to the stability of the protein. The stability of the protein globule is heavily dependent on how well the hydrophobic residues are buried and how the hydrophilic residues are oriented towards the solvent. Proteins with mutations that expose hydrophobic residues to the solvent often lose their stability, leading to misfolding or aggregation. The problem of protein aggregation occurs when multiple protein molecules expose hydrophobic residues and these residues interact with each other instead of being buried within individual protein cores. This can lead to the formation of non-functional aggregates, which is a phenomenon associated with various diseases such as Alzheimer's and Parkinson's. From an evolutionary perspective, proteins have evolved sequences in which the distribution of hydrophobic and hydrophilic residues is optimized for proper folding. Natural selection favors sequences that lead to stable, functional protein globules where hydrophobic residues are efficiently packed inside the protein and hydrophilic residues are exposed to the environment. With this, I am ending the video. Thank you.